Very good afternoon, students. So yes, we are back to discuss APEAPCET paper of May 19 shift to second segment. Okay. Yesterday we have dealt with first 20 questions and today we are going to deal with the rest 20 questions. Okay. And in this session we are going to discuss the concepts as well, memory hacks and mnemonics as well. By solving each and every question, we are going to tackle from which chapter that question has come from and what is the concept relevant to that chapter. Okay, and what more questions can we expect from that segment of the concept. This and all we are going to discuss. This is highly useful and beneficial to all the students who are writing APEAPCET, both AP and TG in the year 2026. Okay, and this is also helpful for the students who are going to write for NEET also. So yes, to all the students who have just landed to 12th standard and to long term students also, this is really helpful. So yes, let's get started. So look into this question, ma. This is from enzymes and mineral nutrition as well. Okay, see here what chapters are involved with respect to this question: biomolecules and enzymes as well. As such, if you see nucleic acid structure, okay, nucleic acid structure, to build a nucleic acid, what enzymes are required to, like DNA polymerases, RNA polymerases, right, all these are required, right. Nucleic acid structure, that is DNA, to build that we need to synthesize a new DNA strand we require, you know, DNA polymerases, RNA polymerases, and in replication topoisomerases are also required. Topoisomerases. So all these are synthesized from what? From magnesium. Magnesium is a chief component for all these enzymes. So yes, for nucleic acid structure, magnesium is very important. Synthesis of nucleic acid structure. Okay. For the nucleic acid structure, phosphorus is present in the nucleotide. We all know nucleotide has what? Sugar, phosphate group and nitrogen base. So yes, phosphorus is present in the nucleic acid structure, and for the synthesis of nucleic acid, DNA polymerase, RNA polymerase and topoisomerases are required. Next, stabilize protein structures. Protein structure can be stabilized by what, beta? Sulfur bridges, disulfide bridges, okay, disulfide bonds. So here the element we have to choose is sulfur. And catalase is related to what? Iron synthesis. So yes, here what options we have to choose, ma? A is 4 and B will be, just a second, 1, B will be 1, and C, stabilized protein structure, that is sulfur, and catalase iron, D is 3. This will be the option, and look into the chapters and the combination of the topics which he has asked for the question. Second question, you see, this is from nitrogen fixation. Okay, this is from what topic, ma? Nitrogen fixation, from the chapter mineral nutrition. So here from ammonia during the process of nitrification, first ammonia is converted to nitrite and then it is done to nitrate. Okay, this will be the flow of reaction, and for the conversion of ammonia to nitrate, what are the bacteria involved? What are the bacteria involved for the oxidation over here? Nitrococcus, and here it will be Nitrosomonas and Nitrococcus. So here, to oxidize ammonia to nitrite, what bacteria is required? Nitrococcus. Fine. Next, choose the incorrect statement from the following he has asked. And this is from the classes of enzymes. This is from what? Different classes of enzymes. We have 6 classes of enzymes, right? Like oxidoreductases, hydrolases, liases, ligases, isomerases. These are 6 classes of enzymes. From that the question is asked directly. Ligases will link carbon oxide, CS, carbon nitrogen bonds, true. Ligases will link, that is correct. Second, hydrolases will catalyze halides and wrong. Hydrolases will lead to what? Hydrolysis of ester bonds, might be ester bonds or aldehyde bonds, ketone bonds. What? Hydrolysis of those bonds are with respect to the enzymes hydrolases. So this is wrong. Next, isomerases catalyze transfer of the group between pair of substrates. No, isomerases are with respect to same substrates, which gives rise to isomerization of reaction. Next question, if you see, ma, just a second. Yeah, assertion reason type of question. Decrease in proton number in stroma and accumulation of protons in the lumen creates proton gradient across the membrane, he is asking. So this is from the chapter photosynthesis. This is from the chapter photosynthesis and from the topic chemiosmotic hypothesis. Chemiosmotic hypothesis. So once let us recall the concept, wherein if this is the thylakoid membrane and this is the thylakoid lumen. Okay. So in thylakoid lumen, because of 3 events, one is photolysis of water, second is from the Z scheme, okay, from the Z scheme, from plastohydroquinone, hydrogen is being transferred from stroma to lumen, and from third event, that is because of the last terminal electron acceptor NADP plus is converted to what? NADPH. Okay, that is converted to NADPH, whereby protons which are present in stroma are utilized for dehydration. Oh, sorry, they are utilized to add the hydrogen group to NADP plus and form NADPH there. So because of these 3 events, what happens beta? Proton concentration is increased in the thylakoid lumen and proton concentration is decreased in stroma. Okay, proton concentration is decreased in stroma and increased in the thylakoid lumen, because of which proton gradient is generated, and then by ATP synthase, that is F0 F1 pump. Okay, ATP synthase F0 F1 pump, 4 protons are being released to the stroma and this leads to the formation of ATP. This is what we have learned from chemiosmotic hypothesis, right? Now if you look into the assertion, decrease in proton number in stroma and accumulation of more protons in the lumen creates proton gradient across the membrane, that is correct. Next, in reason you see, energy is released by breakdown of proton gradient by ATP and also synthesis of ATP. So here if you see, both assertion and reason are correct, and in reason it is clearly explaining how the energy is released and why the proton gradient is happening, and at the end what synthesis is being happening, ATP. So here what we have to choose beta? Assertion and reason are true and reason is the correct explanation of assertion. Clear? Next. Here you see, this is from dark reactions or light independent reactions from the same chapter. But look into it how they have asked. The Hutchinson-Slack pathway. Hutchinson-Slack pathway means what pathway? C4, right? Hutchinson-Slack pathway is nothing but C4 pathway. Mesophyll cells lack rubisco. Okay, in C4 plants, mesophyll cells will lack rubisco, that is true. Bundle sheath cells will have rubisco in C4 plants, right? Next, PEP carboxylase is present in bundle sheath of chloroplast. Wrong. In bundle sheath cells of the chloroplast, what is present? Rubisco is present. Oxaloacetic acid is formed in mesophyll cells only, that is correct. Next, phosphoenol pyruvate is primary carbon dioxide acceptor and which is present in the chloroplast of bundle sheath cells. Wrong. This is present in the chloroplast of mesophyll cells. This is present in the chloroplast of mesophyll cells. You will clearly understand this once you look into here. In mesophyll cells, what is the primary acceptor of carbon dioxide? Carbon dioxide, after getting into the mesophyll cells, it gets converted into hydrogen carbonate by carbonic anhydrase enzyme. This is primarily accepted by 3-carbon compound, that is phosphoenol pyruvate, and the enzyme which is catalyzing this is PEPcase, phosphoenol pyruvate carboxylase. Okay, and from here hydrogen carbonate, phosphoenol pyruvate, in the presence of PEPcase, this forms 4-carbon compound, that is oxaloacetic acid, which is being pumped to bundle sheath cells. In the chloroplast of bundle sheath cells, which has a granule chloroplast, it has rubisco. Okay, it has rubisco, and decarboxylation happens, which releases carbon dioxide and 3-carbon pyruvate. Okay, this 3-carbon is pumped back to the mesophyll cells. Carbon dioxide will run C3 cycle. This is the gist of C4 plants, right? Hutch and Slack pathway. I have given you the gist without bioenergetics. I have given you how much ever is required to the question. That is what I have explained you. So here you should be clear that phosphoenol pyruvate and PEPcase are present in what cells beta? Mesophyll cells of C4 plants, and rubisco, RUBP are present in the bundle sheath cells of the C4 plants. Clear? So now here what are the correct statements? In statement based questions, what we have to primarily look into, whether they are asking us the correct statement or incorrect statement. Correct he has asked, so 1 and 3 will be the answer. Next, arrange the intermediate compounds of Krebs cycle based on number of carbons in ascending order. Okay, based on the ascending order we have to give actually. So alpha ketoglutaric acid, which is 5 carbon, right? This is from TCA cycle. TCA cycle or Krebs cycle, where we can encounter 2 carbons, 4 carbon, 5 carbon and 6 carbon. Right. So now if you see, alpha ketoglutaric acid is 5 carbon compound, acetyl CoA is 2 carbon compound, citric acid is 6 carbon compound. Yes or no beta? Now you look into it carefully. Acetyl CoA, he asked us to identify this in ascending order. Acetyl CoA 2 carbon, 6 carbon, 5. Wrong, we cannot go with this. First alpha ketoglutaric acid 5 carbon, citric acid 6 carbon, acetyl CoA, we cannot go with this 2. And then if we see acetyl CoA, which is 2 carbon, alpha ketoglutaric acid and citric acid. This is in the ascending order. So yes, this will be the correct answer. And next question you look into, again match the following question. This is from the chapter Plant Growth and Development. Okay, this is from Plant Growth and Development. From that, from what segment the question is asked? The question is asked from plant growth regulators. Okay, so now you clearly see kinetin, which is artificial form of cytokinin. Okay, this is synthetic cytokinin. Kinetin is what? Synthetic cytokinin. So kinetin, which is synthetic cytokinin, these are what derivatives? These are from adenine derivatives, or you can say purine derivatives. Okay, purine derivatives or adenine derivatives. In your in your academy or NCERT, one nice term is given, that is N6-furfuryl aminopurine. Okay, N6-furfuryl aminopurine. Clear? Next, Abscisic acid, these are carotenoid derivatives. Okay. Gibberellic acid, these are terpene derivatives. Indole acetic acid, these are indole compounds. So, this is the match the following type of question. Along with the composition, you should be clear with respect to the precursors as well. Just go through that. Next, choose the correct series of bacteria that cause tetanus, diphtheria, crown gall, blight. Okay, we have to choose correctly with respect to the diseases. This is from the chapter bacteria and viruses, both the chapters included and asked beta. So, here you see, first he has asked tetanus. Clostridium tetani is caused from what? Clostridium. Next, diphtheria, Corynebacterium diphtheria. And third disease is about, crown gall. Crown gall disease is caused by Agrobacterium tumefaciens. And next, bacterial blight is caused by what? Xanthomonas. So, this is from microbiology, bacteria and viruses. And then number of nucleotides in viroids. In viroids, the number of nucleotides include 300 to 400, and the molecular weight of viroids is less than virus, because virus is a composition of both genetic material and protein coat. Okay, virus is the composition of both genetic material and protein coat. Genetic material and protein coat. Fine. Next, if you see, the phenomena of a single gene product may produce more than one effect, so that single gene may be related to more than one character. One gene giving the expression of multiple characters is what comes under what concept? Pleiotropy. Okay, pleiotropy, which explains single gene showing multiple effects. Best examples under pleiotropy includes what? Phenylketonuria disease, one of the best example under pleiotropy, where single gene is showing multiple effects. Okay, this is the exception or deviation from Mendel's laws, right? So, directly it is from the definition of the pleiotropy itself. So, it is very simple question. This is from Principles of Inheritance Genetics. Next, mutation is a phenomena of, this is from Malbio. Okay, mutation is a phenomena of what? Three will come: alteration of genes, or variation in DNA, or heritable change in both phenotype or genotype in an organism. All these three will lead to what? Mutation itself. Next, this question, if you look into this is from molecular biology, Malbio, wherein purines and cytosine are common in DNA and RNA. Yes, purines and cytosine, they are common in DNA and RNA, that is correct itself. Next, thymine is present in RNA, wrong. The nitrogen bases which are in RNA and DNA, we will look into once. The nitrogen bases which are in DNA includes adenine, thymine, guanine and cytosine, where adenine and guanine are what? Purines. Thymine and cytosine are what? Pyrimidines. RNA includes what nitrogen bases? Adenine, uracil, guanine and cytosine. Adenine and guanine here are purines, uracil and cytosine are what? Pyrimidines. Clear? So, yes, thymine is not present in RNA. Third statement, if you see beta, uracil is present in DNA at the place of thymine. Wrong, uracil is present in what? RNA. Adenine and guanine are purines. True, adenine and guanine are purines. In question, you clearly see whether he is asking you the correct statements or incorrect statement. Incorrect statements are asked. So, what we have to go through? 2 and 3 will be the answer. Next, assertion reason question. In both AP, APECET and TG APECET, most of the questions you get are assertion reason and statement based questions only, which test your subject level to the peaks. Okay, so practice and exercise in that way only beta. Assertion and reason, if you see, assertion, in prokaryotes the DNA in nucleoid is organized in large loops held by proteins. In prokaryotes, if you have to consider, prokaryotes will have two genes, right? Two, one is genomic DNA and the other is plasmid DNA, okay, genomic DNA and plasmid DNA. So, genomic DNA is placed at one position without covered by any membrane, non-membrane bound. This region is called as what? Nucleoid. So, this is placed at one position because DNA is held by some positively charged proteins. But this positively charged proteins in prokaryotes, they are not histone proteins. They are not what? Histone proteins. They are just combined with some positively charged proteins like polyamines. Reason, if you see, in prokaryotes a negatively charged DNA molecule held with positively charged proteins, correct. Here assertion and reason both are true, and reason is relevant to assertion, which is explaining the assertion. Okay, reason is explaining the assertion in a positive note. So, what option we have to choose into? Option 1. This is the question from biotechnology. Zoology sir will be dealing this beta. Here you see, this is from strategies. Protein toxin produced by Bacillus thuringiensis that kill corn borer. Always do remember, from corn borer, okay, corn borer is caused by which toxin will kill, which protein toxin will kill is Cry1AB. Cry1AC and Cry2AB are, this will kill cotton bollworms. This will kill cotton bollworms. Clear? Next question. Choose the correct statement from the following. This is directly from strategies. All the statements are given in a direct note beta. So, here if you see, validity of genetically modified research and safety introduction of genetically modified organisms have to be given by GEAC, correct. Molecular farming means utilization of plants and biofactories for obtaining commercially useful products. Yes, this is also true. Next, Basmati rice variety is transgenic plant with improved nutritional value. No. Flower saver is suitable for food storage technology, correct. Flower saver, which is the GMO of what? Tomato. Advanced variety. So, here what statements we have to pick? Correct statements, which includes 1, 2 and 4. Next, the following hybridization crop variety is not an insect resistant variety. Okay, this is not an insect resistant variety. This is from the direct tables of that strategies topic, that is Pusa Komal. Okay, that is option 3. Next, assertion reason type, again this is from microbes in human welfare. Microbes in human welfare chapter. So, here if you see my assertion, Propionibacterium shermani is responsible for large holes in Swiss cheese, correct. Next, in reason if you see, characteristic texture, flavor, taste of cheese is specific for microorganisms. True, characteristic texture, flavor and taste of cheese is specific for microorganisms. Here both assertion and reason are true, and reason is clearly explaining the assertion. Okay, like why large holes are formed only by the bacteria Propionibacterium shermani. Okay, so here A and R are true, and R is the correct explanation of A. Next, choose the correct statements among the following. This is also from the same chapter, microbes. Microbes in human welfare. So, here you see this is the last question from this paper. So, streptokinase is produced by streptococcus, true, streptokinase is produced by streptococcus. Saccharomyces cerevisiae is used for commercial production of ethanol, true. Saccharomyces cerevisiae yeast is used because yeast will conduct what? Anaerobic respiration, that too alcoholic fermentation. Alcoholic fermentation. Clear? Next, cyclosporin A, blood cholesterol lowering agent produced by Trichoderma polysporum. This is correct statement only, but I don't know why it is not being answered properly. This is also correct statement. Cyclosporin A is produced, which is a blood cholesterol lowering agent, is produced by Trichoderma polysporum. Just a second, cyclosporin A is produced by Trichoderma polysporum, that is true, but it is not blood cholesterol lowering agent. Okay, that is why this is wrong. Next, statins are produced by yeast. This is true. Okay, statins are produced by yeast only. So now here we have to go for what? Correct statements. Okay, statement based question where he is asking you to identify correct statements. So here what statements we have to look into, ma? 1 is correct, streptokinase produced by the bacterium Streptococcus, and yeast is commercially producing ethanol. Cyclosporin A is produced from Trichoderma polysporum, that is true, but it is not the blood cholesterol lowering agent. Statins are produced by yeast, true. So here what statements we have to opt? 1, 2 and 4. Okay. So option 4 will be the correct answer. Okay. So tomorrow we are going to look into the last paper of APEAPCET, which will take again 2 more classes. So yes, stay tuned and thank you. To view more such videos on questionnaire sessions, and in this questionnaire session we are also planning you to give top 1000 questions from all the concepts of botany. Okay, top 1000 questions, like I will tell you what chapters are being included under what concepts. So stay tuned students, at the same time at sharp 2 p.m. each day we will have the questionnaire session. Okay, thank you.